Hello, I am Deep Dutta. I'm an endocrinologist based in Dwarka, New Delhi, and today we shall be discussing how to do meta-analysis. So trust me, we do meta-analysis using softwares. We have a lot of different softwares and they are very easy to use. We just need to give some time practice on them. Today we shall be learning how to do meta-analysis using the Revman 5 software. So we will start from basics, starting from how we download this software and to how do we go about doing the meta-analysis. So before doing a meta-analysis, we should be clear what are we trying to achieve. We should always try to do something which is novel, something which is new. There's no point of repeating something which has already been done, or else it will have very little clinical significance. So meta-analysis are a very objective way of looking at data from different randomized controlled trials. So we know we do randomized controlled trials to find whether a particular treatment modality, a particular intervention, maybe a particular medication, whether it really works or not in a particular disease state. So you may have different randomized controlled trials published from different parts of the globe, and not all of them will give same results. So some may give very good results, some may give an average so-so results, and some may give maybe totally opposite results. So as a treating physician, many times we are bogged down which of these studies should we believe or whom not to believe. So meta-analysis is a very objective, quantitative view of analyzing data. You pool the data, the results of all the different RCTs, and then you get a net output which tells us whether that particular treatment or intervention really works. What the meta-analysis software does, that once it analyzes the data from different RCTs, it looks at the strength of the data. So studies which have greater number of patients, they are given more weightage. Studies, if you're looking at, say, a reduction in HbA1c for any diabetes medication, so it looks not only at the mean values, it looks at the spread of the data. It looks at the standard deviation. So for any observation, greater the standard deviation, greater the spread out of the data, lesser is the strength of the data. So, so let's now go into a particular molecule. Say we have published a meta-analysis on evaluating the role of Gemigliptin in the treatment of type 2 diabetes. So I'll just brush through again how did we go about doing this, what made us think to do this meta-analysis. So I went to PubMed. I did a search on Gemigliptin to see what all literature is there, and we find here that we have nearly 85 research articles on Public Library of Medicine on Gemigliptin on different clinical scenarios. Now I want to be even more specific, so I want to see how many of these articles had the word Gemigliptin in their titles. They'll be more specific. So we are down to 58 different articles. See here that we have lot of different articles have been published. Many of them may be RCTs, many of them are simple case control studies. So I would like to know how many of them RCTs are published regarding the use of Gemigliptin. So in PubMed, you have the left side column. You can click randomized controlled trials, and you see there are 22 different RCTs. So if you want to do a meta-analysis, we should do a literature search to see whether any RCTs have been published or not. If there are no RCTs, you cannot do a meta-analysis. Ideally, you should have at least 3 different RCTs published on that particular intervention, topic, or medication to be able to do a reasonably good meta-analysis. So what we did, that we found all these RCTs from here. We downloaded them one by one into our laptop. We made a separate folder called the Gemigliptin meta-analysis, and we sorted them by the name of the authors and the year of publication so that we can go back to them and review them whenever we want to. Trust me, it's a highly labor-intensive job. So you need to have a team of at least 3 to 4 of your colleagues, or to share the work so that you can finish the work in a stipulated time, maybe 1 or 2 months. Yes, it takes many months to do a meta-analysis. It can't be done in a day or 2. So just like we search in Public Library of Medicine or PubMed, you can do search in Medline, you can do search in Embase, you can do search in Google Scholar. The idea is not to miss any particular RCT, because not all of them are indexed in only one particular software platform. So you can go to Google Scholar, you can do a search of Gemigliptin, and you will get all the articles. You have to search it. It's a good practice to repeat the search by 2 or more of your team members, because if I do a search, I may miss some article. It's likely that will be picked up by some of my other team members. So once we have searched, we have downloaded the articles. The key point here is to find out, has any meta-analysis already been published on this topic? So the PubMed is very good. It gives an option of selecting only meta-analysis. And here it shows that as of today, there are 2 meta-analyses published already on the use of Gemigliptin in type 2 diabetes. So, but when we did this meta-analysis, there were none. So that's why we went ahead and did it, and we published this meta-analysis. It's available in the public domain. So if you look at the meta-analysis, just the abstract, if you see, it mentions why we did this study, why we did this meta-analysis. We did because there was no data available at that point of time. Then it mentions how we went about looking for the different studies, different RCTs, different platforms. We should be very clear what are we trying to achieve. Our primary outcome and secondary outcome should be very clear. You should have a consensus with your team members. Like for the anti-diabetic medication, our primary outcome was the quantum of reduction in HbA1c. Was it really good as compared to the control group, which received either a placebo or maybe another anti-diabetes medication? And here you get a result here. If you see here, it always talks of mean differences. So mean differences is the average reduction of a particular parameter. Here in this case, the primary outcome was HbA1c, and it always gives an outcome in the 95% confidence interval. It also talks of a value called the I-square. So all meta-analysis will have an I-square value. It talks of the degree of heterogeneity, that is the different studies, how they are similar to each other, how different they are. A 0 I-square means no heterogeneity. 100% I-square means they are totally heterogeneous. All the different studies are talking of really different things here. So whenever you look at the mean difference studies with low I-square values, the strength of data is good. Greater the heterogeneity, lesser is the strength of data. So we are really not sure whether it will be really replicable in our day-to-day practice. Also, it talks of odds ratio with regards to certain dichotomous events, or maybe even for adverse events are reported as hazards ratio or odds ratio. So now let's go to the Revman software. How do we get the Revman software? You can do a Google search. You can just go to the simple Google and you can do a search Revman. And it comes here directly, the Cochrane. The Revman software was developed by the Cochrane Group. They are a very reputed group. They publish a large number of meta-analysis. They are very good at doing huge meta-analysis. So this website from which we can download for academic purpose, it's free to use, it's non chargeable, and you can install in your laptop or your desktop. And not only that, they give a lot of learning material. So there's a lot of learning material here. You can go and read them, how to use this software. They are very simple, easy to read, very simple language. It just needs some time to go through it. So they have even handbooks, beautiful handbooks written on how do we do systematic reviews and meta-analysis, how we do grading of the results. Grading is a totally different thing we will discuss some other day. So what I did for the first time, I downloaded this software and I saved it on my desktop. So let's now go to the Revman software. So I've opened the Revman software. This is the screen. If you see here, this is how it looks when you open. Of course, everything was blind. I have already opened the Revman software, on which we did the Gemigliptin meta-analysis. So the first thing you do here is you write down your topic. So our topic was to find out the efficacy and safety of a novel dipeptidase 4 inhibitor, which is Gemigliptin in type 2 diabetes, because there was no data published at that point of time. You do this and you click the save. Keep on saving always, or you don't want to lose data. The next thing to do is you add your team members. We have already 3 team members. There's other author also. You can add. You have the add author option here. You can add Here you can put whatever name, say you can add Mr. XYZ, the first name and the last name will be ABC, and you finish it, and the name of the author keeps on getting added here. So once you have decided your team members, you have added the author's name. Now let us look on the other side. Here many things are there. There's a main text. So the beauty of Revman is that you can write your whole meta-analysis on the Revman software. You can put your background, you can put your main objectives of the abstract. Not only that, you can do the main text also. You can write the background, you can write the introduction of the main text of the manuscript. So I have done by conveying on the Word document. You can as well do it here in the Revman also. It's totally personal choice. The next important heading is the tables. Of course, we're not making tables now. We need to enter data. So we'll come to it later. The next most important thing is the studies. What studies you have included? So you go to the studies heading. You see the reference to the studies, and you see something which comes always as the included studies or the excluded studies. So we have downloaded the articles, and you have made in folders. So some of the studies may fulfill your inclusion criteria. Some of the studies may not fulfill your inclusion criteria. So our criteria was that the primary objective was that the study should have, the RCT should provide data on HbA1c reduction. So if you have an RCT which talks only of fasting and postprandial blood glucose, which doesn't talk of HbA1c, so that is not included in our study. So here you can, if you see here, we have entered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 different RCTs. You can always add more RCTs. You can see, right click on the included study. You can click add study, and you can enter the first author's name. Say it can be, say, Mr. ABC, and you put the year of publication. Say it was published in 2022, and you keep on going to next. You keep on going. The year comes here automatically, and you do nothing. You finish it and see. It arranges the name of all the authors in alphabetical order. So the A, B, C comes above because A by the alphabetical order. So you can keep adding more. If you feel you have entered something wrong, you can right click on it, and you can even delete it also, that the study gets deleted. If you have entered something wrongly, you can always edit it, and don't forget to save it. Similarly, you can go to the list of excluded studies, and you can see the name here. One study was not fulfilling our inclusion criteria, so we entered here. Click here, add study, and keep on adding just like we did previously. So once you have entered all the studies, so before doing this, a lot of background work goes into that. Your team members and you, each one of you will have to read each of the RCTs to see whether they fulfill your inclusion criteria or not. So once you have entered the studies, now Revman software or any meta-analysis software would like to know the biases associated with each of the studies. So there is a section here. If you go up here, the moment we enter the name of the study, a separate set of columns come for the nature of the bias. So while we do meta-analysis, these are the 3, 4, 5, 6, and 7 main different types of bias. So it wants to know whether the random sequence generation was done, that the patient was allocated randomly or not. We don't want to say that the treating doctor or the patient should know what's happening, right? So greater the degree of blinding, lower is the risk of bias. So here it was a double-blinded study, so you can always select either low risk. You may not be sure, you can select unclear risk. If there was no random sequence generation, you can select high risk here. Since it was a randomized double-blinded placebo-controlled study, we got this information from the manuscript, published manuscript already. I just copied it from there to here. Next bias is the selection bias. Each RCT needs to mention how the randomization was done. Did they use a computer-generated randomized table? Did they do a simple randomization, or did they use a stratified block randomization? This study, this RCT, done by Dr. A.H. and An et al. They did a stratified block randomization. So we got this information again from the manuscript, and hence it became a low risk. Third, it talks of blinding of the participants and personnel. It was a double-blind RCT. Yes, it was low risk. It talks of blinding of outcome assessment. Those who assessed the outcome, the treating doctors did, where they also blinded, and it was yes. So here it also became low risk or not high risk. Then we talk of something called the attrition bias. It means that, say, we have randomized a certain number of people in this study, say 290 people who have randomized. This wants to rule out loss of patients to follow. As a rule, any study which uses more than 10% of the patients which were initially included is considered to have a high attrition, and then there it becomes a high attrition bias. But if you see here, 219 patients were randomized, of which the final analysis was done from 216 patients. We just lost 3 patients. The attrition rate was very low, only 0.01%. So hence the attrition bias here also low risk. We selected low risk here. Then we talk of something called the reporting bias, by which the study, the RCT tells us in the beginning what are their primary and secondary endpoints. We need to go to the result section and see whether they have actually elaborated on each of the primary and secondary endpoints. If they missed out on reporting of some of the endpoints, whatever may be the reason, we call it a selective reporting, and we call it the selective reporting bias. So in this study, all the pre-specified outcomes were reported. So here also we selected a low risk bias. Now the other bias looks at the overall study. Was the study funded by a pharmaceutical organization, where one or more authors were from the pharmaceutical organization involved in the generation or development of this molecule? So this study was funded by LG Life Sciences, and we know even one of the authors was an employee of LG Life Sciences, and we know LG Life Sciences has a major role in the development of gemigliptin molecule. So here the bias was high risk. So like this, you have to read each of the RCTs carefully, get their information, and you keep entering. So we did for the 3rd study, we did for the 4th study here, we did for the 5th study, and you keep on doing it. So if you see here, one of the studies by Dr. Park, it was a randomized controlled trial, but it was an open label trial. So the blinding of participants here became high risk and not low risk. Even the outcome assessors were also high risk, but it's an open label trial. And the manuscript did not mention how did they go about doing the randomization. So we're really not sure. So we ended up selecting here unclear risk, not low risk, not high risk, but unclear risk. And again, it was funded by LG Life Sciences. The other bias was high. So like this, we do it for all the 11 RCTs which was included in this meta-analysis. Once we have done that, you can generate a holistic, nice looking figure which tells you the overall bias of the meta-analysis of the studies which have been included. You go to the figure section, you can always go add a figure, and add a figure, it talks of risk of bias summary. Once you click next and you finish it, you get a nice looking curve like this. So now this talks of all the bias. Green means low risk, yellow means the unclear risk, and red is the high risk. If you see, for majority, all the study, the random sequence generation was very good. There was no attrition of data. There was no reporting bias in all the 11 RCTs. But if you see the other bias, which is funding by pharma organization, was very high. Only one study was not funded by the pharmaceutical organization. So much information we are getting from this beautiful curve, with this graph. And few studies had a lot of unclear risk also. You can click on it and you can save it. So if you see here, you click on the save sign, you can save it in your folder. You can save it as an SVG file, or you can save it as a PNG file, or even a PDF file. I typically save as SVG file because the clarity of the figure is the best in SVG file. You may save it as a PNG file also on whatever folder you have made for your meta-analysis. Similarly, the other figure which we would like to generate is we go to add figure. We want to see the risk of bias graph. So now you get a similar information in a slightly different form. You get the information like this. You can say that 100% of the study had low selection bias, attrition reporting bias, but 20% of the studies had high performance and detection bias. So this figure also, you can click on it and you can save it in your folder for the meta-analysis. You can keep on doing it. So once we have done this work, then we would like to, the next most important thing is the data entry. So data entry of different studies. So where do we do that? So for you go here, the data analysis segment, and you click here. So there we have already, you may have different doses of different drugs, right? So we already have a heading which compared the full dose of gemigliptin 50 milligrams compared to controls. We had a subheading where we compare 25 milligrams to controls. You may choose to compare, you may have as many comparisons as you want. So if I want to do a third comparison, say I want to compare People who were receiving a very low dosage, 10 milligrams of gemigliptin versus control. It doesn't happen in the real world, but we are just doing it for the learning purpose. And we do a next, and we finish. And what do we get here? Give it some time. If you see here on the left side, you get a third set of comparison. Don't forget to save whatever your work you have done. And you can keep on adding variables. So let us go to the full dose gemigliptin versus control. I've clicked here. So you see what all analysis we have done already. We have compared the HbA1c reduction at 12 weeks, at 24 weeks. We have compared the fasting glucose, the postprandial glucose, the glucose exertion, the total and severe adverse events, the occurrence of symptomatic and asymptomatic hypoglycemias, for the lipid parameters, the insulin resistance parameters, and we have done it separately for the active control group. When the control group was another anti-diabetic medication, or we had done it for the passive control group separately, where the control group was placebo. So how did we generate so many of these variables? So we have generated this heading. You can again right click on this. You can add an outcome. Say I want to add an outcome of HbA1c reduction at say 54 weeks. So before going ahead, we need to tell the Revman what sort of analysis we are doing. Now, dichotomous means we know yes or no, true or false, but HbA1c is a continuous variable. So we change it from dichotomous to continuous for any variable. For the first time which we do it, Revman will always ask us this question. Next, we go to what are we doing? What are we trying to look at? We are looking at HbA1c reduction at what? 54 weeks. And in what group? We are looking in the active control group, not the passive. So we go to the next. What sort of analysis we are doing? Traditionally, by convention, we always do a random effect analysis. Why so? Because we have taken data from so many 11 different RCTs from different part of the globes, different ethnic groups. So they are not a homogeneous group of individuals. So in them, we always do a random effect analysis. Fixed effect analysis is very rarely done when all the RCTs are done in a similar group of population, similar age group of patients, a very similar group of patients. So, which is very rare in medical science, so we almost always end up doing random effect analysis. We had talked of I-square value or the heterogeneity. Whenever the I-square is more than 50, we always do a random effect analysis. We are looking at the mean difference of HbA1c is perfectly fine. We are going to enter the mean values. And we are looking at the 95% confidence interval because that's how most of the medical literature are reported. You may choose to do a 90% confidence interval or 99, but since all the RCTs have the data in terms of 95% CI, we do a 95% CI in our meta-analysis also. And the left side of the graph favors the experimental agent here is gemigliptin. The right side of the graph favors the control group. So we enter, and what's the unit? You can put the unit if you have. For HbA1c it is percent. And we have entered by study ID, which is the name of the first author and the year of publication. I'm sorry. And you keep on going to next. And here what you do is initially for the initial purpose, nothing. And you finish it here and see it has come down here, the HbA1c 54 weeks. So we have already made it twice. If you have some error, you can always go back and delete it. If you've done it twice. And don't forget to save it whenever you make any change in your Revman file for your particular meta-analysis. So let us see what we do subsequently. We click on it here. You see, I have added data of 3 studies. We had 11 studies, but we have entered only 3 studies because only 3 of the 11 studies had data for HbA1c reduction at 12 weeks. Let us look at 24 weeks. Only 2 studies had data for HbA1c reduction for 24 weeks. So not all studies will have all the different parameter outcomes. Different study looks at things differently. We look at total adverse events. You see all the 11 studies are here because almost all the 11 studies reported the total adverse events. So how do we keep add studies? We have entered the parameter like say HbA1c. 12 weeks here, you do a right click on it. You say add a study data. So it will show what all 3 studies we have already added and we have not added. So if you think any of these studies has data on HbA1c reduction at 12 weeks, you can jolly well click here, click it, and you can click finish. And it comes here. If you see here, since the study does not have, I am just deleting it off. because we don't have data from this study for HbA1c reduction for 12 weeks. Now you can select multiple studies at a time. So let us repeat it again. Right click, add study data, and you want to add multiple studies. You can click the control on your laptop. You can keep selecting one study, two study, three, whatever studies you think they have, and you click finish, and all four of them comes here. If you see here, they've come here, and don't forget to save it, right? And similarly, you keep on adding like this, and you keep on going forward. So once you have done that, the next step would be here is to enter the different variables. So as you see here, we have entered the information for the different RCTs. The study by Kwok et al. Published in 2020 from Park 2017. A and B. Now you must be wondering what is A and B. Really, you may have two RCTs which are published by the same author or in the same year, but they're different RCTs. Then you can number them A and B. So that helps really in differentiating. So these two studies we just randomly added. So I'm just deleting it off. And I think, let us see here. We have added two just for the sake to show how we add study here. We are deleting them off again because they don't have the data. Let me save it. And next, what we did, if you see here, you see there's a mean value and the standard deviation. So we go back to each of the RCTs, we find what was the mean reduction by of HbA1c in the study, and we enter here. We enter the standard deviation, we enter the number of people in the Gemigliptin group, and the same thing we do for the control group for each of the three RCTs which were here. And once you have done all that entry, you click here, there's something called the forest plot. I click here and you see a nice, beautiful looking forest plot in front of you, what we are trying to achieve here. Now, how do we read this forest plot? So now it talks of the overall outcome here below, and it talks of the outcome of three studies. If you see here, and it also gives a weightage. The Revman Software has given nearly 99% weightage to the first study by Kwok because it had the maximum number of people evaluated. And if you see, the standard deviation was very low, so it gave the maximum weightage. For the second and third study, the standard deviation was so high. So greater the standard deviation, we discussed some time back, lesser is the weightage given by the software. And here the final outcome, if you see, there is a mean reduction of nearly 0.3% HbA1c with Gemigliptin at 12 weeks of follow-up, and is statistically significant because in the best case scenario, it is as low as minus 0.35% reduction. And in the worst case scenario, it is as still a significantly low at minus 2.5% reduction. So the p-value here is less than 0.05, which is significant. And if you look at the I-square, the heterogeneity is 0 because all the three studies are towards the left side of the graph. So lower the I-square, greater is the strength of your observation. And you click the save option, and you can save this figure as a SVG file or a PNG file or a PDF file, whatever you want, on the folder for your meta-analysis. So since I've already saved it, so I'm not saving it again. And similarly, you do it for 24 weeks. So now let us look at total adverse events. We looked at the number of events reported in each of the RCTs. So there's a lot of hard work. Go to each of the RCTs, get the values, enter the correctly. If you enter it wrongly, the Revman will give you wrong results. So it's a good practice to always, two or more authors should cross-check the data entry done so that you pick up the errors done. So if you see here, if you look at now, total adverse events are dichotomous events. They're not continuous events. So once we look at the forest plot here, you get a value in odds ratio or the hazards ratio. Now, if you look here, some of the studies that going towards the control, favoring control, some of the studies is favoring the Gemigliptin. So hence the I-square is not 0. There is some heterogeneity. It is 35%. So by convention, we say any heterogeneity less than 60% is considered low. 60 to 90 we call moderate heterogeneity. Anything more than 90% is called a very high heterogeneity, and we really need to take those results with a pinch of salt. And again, we click here save. Since we have entered the data, you can save the forest plot for your total adverse events, and you keep on doing such entry analysis for whatever variables, how much variables you want to take in your meta-analysis, and you keep on saving each of the different forest plots in your folder. So once we have done that, I think we can close the Revman software here. I'll stop sharing the screen here. And let us go to the folder where we have saved so much of information. So let us go to the folder. Yes, this is the folder I'm sharing with you. This was a Revman file which had all the data. So we had nicely made a folder for the RCTs which we had included in our meta-analysis. If you see here, we have saved them in terms of the molecule name and the name of the first author and the year of publication, because so that even my co-authors, when they're going to review it, it's easier for them to go back. Never forget to download the supplementary material, because there's so much information in each of the RCTs. It is impossible to incorporate all of them in the main text. Supplementary materials are a treasure trove of lot of vital information which we have to include in our meta-analysis. So always download the supplementary material and take them into your analysis also. Once you have done that, see, if you see here, I have made a folder for manuscript submission when our team was preparing the manuscript. And if you see here, we saved all the different figures. So here is the folder of isolated figures, if you see. So we had painstakingly downloaded each of the forest plots here, if you see here, for different outcomes for HbA1c reduction at 12 weeks, 24 weeks, in the active control group, in the placebo group, or the passive, or the PCG, or the placebo control group. So now we have saved so many figures. If you see here, you can't submit so many figures in your article, right? So you have to merge the figures. You can use a Microsoft Paint software. You can use any software to merge these figures. So similar figures are merged together. If you see, and you get something, you can always make your consolidated figures. If you see here, the consolidated figures, if you see here, they look, you save a lot of space. So that this is a consolidated figure of HbA1c reduction at 12 and 24 weeks in the active and the placebo control group. So similarly, we made a lot of consolidated figures here. And then finally, you end up writing your manuscript. Lastly, we will look at how we write the manuscript of a meta-analysis. So we have already done for this Gemigliptin meta-analysis. So let's open the document file. Yeah. So if you see here, you can see the manuscript here. Let me share the manuscript again to make it. Yeah. So you can see the manuscript here, and the most important thing is that we would have written the protocol. We were already clear with our primary aims and objectives. We were already clear with the inclusion criteria. We have already entered the data in the Revman file. We have downloaded the forest plots, and we have merged them into the main forest plots, because you can't submit 18 to 20 forest plots for publication. So you have only 4 or 5 figures, the main forest plots. We bring them together. And when we write the manuscript, first of all, the initial part would be like with regards to Gemigliptin, regarding the current data available, how Gemigliptin works, and in type 2 diabetes. And then we build up to the primary objective, that since there were no meta-analysis published at that point of time evaluating the efficacy and safety of Gemigliptin, we try to assess that in our research study. And then we go about how we did this meta-analysis. We followed the Cochrane methodology, because when you use the Revman software, you're using the Cochrane methodology to do the meta-analysis. And as a good clinical practice, whenever we write a protocol, we should register it at the Prospero. How to register at Prospero, we will discuss some other day. It's again a free registration. We upload our protocol there, and it is freely available for anybody to review and check. So it's a good clinical practice. We follow the PRISMA guidelines regarding how to do meta-analysis. So it's a good thing to read about what does the PRISMA guidelines talk of. Again, they're freely available. We enter here the PICOS criteria. Whenever we do the meta-analysis, we follow the PICOS criteria, where we are very clear what are the exact group of P or the people being studied here. It is people with type 2 diabetes. I is the intervention. The information here is the study molecule, which is the gemigliptin as compared to the controls. And C, it talks of the controls. It can be either active control or like any other diabetes medication here in this scenario, or maybe just a placebo. And the O stands for outcomes. So what are the outcomes we're looking at? Our main outcome here was HbA1c reduction. It can also be fasting glucose, postprandial glucose, the side effect profile of the medication. It can be as long as we want it to happen. And S stands for the study type. So in our case, we have included only randomized controlled trials for doing this meta-analysis. We then state in our manuscript the main primary and secondary objectives. We mention what all database we have searched to get all the RCTs which we have included in our meta-analysis. Then we talk of which of the authors, how many authors were involved in data screening, data searching, getting the RCTs out. And then we talk of how we use the Revman 5 we discussed some time back to find the risk of bias for each of the individual RCTs which we have included here. We then tell that we have presented the analysis, the outcomes of the meta-analysis in terms of mean difference for continuous variables, and we use risk ratios for the dichotomous variables and hazards ratio for the adverse events or mortality. Then we talk of data heterogeneity. We have discussed how the I-square value we get from each of the forest plot. Revman does it and gives us the I-square value. We have discussed how to interpret the I-square values, and we write it in the manuscript. Then in the end, we have got all your results, some of your main outcomes. We would like to know how good are your results. What is the grade of evidence? It is a really good evidence, high grade evidence, or a moderate grade or low grade. So there is a separate website called the Grade Pro GDT where we can find the grading of your analysis. How to do grading is itself a separate topic. We shall discuss some other day. And then you go about writing down the result section where we initially write how many studies you searched for in the PubMed. And like in this case, PubMed, we had found around 83 odd articles. We got some other articles from the Google Scholars. So we had 95 articles to start with, and how we came down to 10 to 11 different articles. And then we cite each of the articles. We talk of what was the control group, was it active control or passive control. We mentioned initially what were the active controls in different studies. We measure about the risk of bias. We had generated the risk of bias graph. And we just mentioned it here, what were the percentage of studies which had the different types of risk of bias. And then we start with the primary outcome. The primary outcome was HbA1c reduction. We talk of both separately for the active control group and passive control group. What was the quantum of HbA1c reduction in the meta-analysis, and whether it was statistically significant or not, and whether the I-square was low or moderate or high. And subsequently keep on adding the secondary outcomes. We present the outcomes for fasting glucose here, the percent of people who had an HbA1c less than 7%, whether the people reaching an HbA1c less than 6.5% was it higher with gemigliptin as compared to controls. Those things are discussed here. And then lastly, but not least, we talk about the safety data. We talk whether the occurrence of total adverse events, severe adverse events, or hypoglycemias, were they similar to the controls or greater than the controls. We present them. As you see here, these are dichotomous variables. So from mean difference, we have now mentioned them here as relative risks, and with 95% confidence intervals, and the p-values, whether it's significant or not, and the I-square values. And lastly, we always mention the figure number if you have mentioned that figure as a part of your manuscript. Here in this case, we did analyze the lipid parameters also. There's a separate section for that. The impact on body weight, the impact on renal parameters. You can choose as many parameters as you want, provided those data are available in the background RCTs which you have included in your meta-analysis. And so on. In the end, you write the discussion, you write what is the key take-home message from meta-analysis, how your meta-analysis will help in improving the clinical practices. You talk about the different heterogeneity in different RCTs which you have included, and what can be the further directions. And with this, you end writing your meta-analysis. Bye-bye.